My name is Thibault Legal from McGill University in Canada, and I'm going to describe my JCB paper entitled CEP104 FAP256 and associated cap complex maintain stability of the ciliary tip. In the lab, we study motile cilia. Motile cilia are cellular projections that carry out multiple functions in the human body. They're important in the trachea to move mucus, and they provide motility to sperm cells, for example. I'm also showing here a video of nasal epithelium cilia that are beating. Cilia are made of a microtubule-based axoneme, and the diagram on the right shows what a cross-section looks like. We see 9 microtubule doublets at the periphery and 2 microtubules in the middle called the central pair. We know the structures and molecular composition of the ciliary tips are different from the rest of the axoneme, but not to what extent. So we used cryo-electron tomography to find out more about it. We first looked at the structure of the central pair and showed that it is very different in the tip. The known projections that are found on the central pair and the rest of the cilium are absent in the tip. We found yet unknown proteins that link microtubules in the ciliary tip. They're indicated by the green arrows here. We also studied cells lacking FAP256, a known ciliary tip protein. First, we found that the cap complex, which is circled in red in wild-type cilia, is absent when FAP256 is missing. We also find that the microtubules of the central pair, C1 and C2, have different lengths when normally they have the same length. Finally, we see that some microtubules in the tip are bent, possibly because the links are absent in this mutant. Using mass spectrometry, we identified the proteins that are missing in FAP256 knockout mutants and identified a new tip protein called CCDC81. Our current model shows that multiple proteins are absent when FAP256 is knocked out, including the links and the cap complex, and their absence leads to beating defects. To conclude, I would like to thank my supervisor, the people in my lab, and my collaborators without whom this work would not have been possible. If you want to find out more about this paper, please visit the JCB website. Thank you for watching.